All right, our last chapter here this semester is going over fungi. So we've talked about animals, we've talked about plants a good bit, a little bit about the prokaryotic cells, now wrapping up with our fungi. These are multicellular organisms, as many eukaryotic cells are. So the ones that have this complex multicellularity, meaning that they have many different cells that become specialized, meaning they have different jobs. So our plants, where we talked about vascular tissue and ground tissue. Animals, of course, we went through lots of different tissues and lots of different cell types, but fungi are the other one. Now, two of the key characteristics that put organisms into this group and differentiate them from simple multicellular organisms like we'd seen with some of the prokaryotic, like the Anabaena or the cyanobacteria, is that these cells specialize, meaning they become differentiated and do a specific job. They can't do anything, but they have one job, like a muscle cell or an adipose cell. So it has a very specific job, and it has intracellular coordination to where it has specific, is a way to do those specific jobs. So a little bit more about that. Again, that specialization requires that different cells use different genes. Now, all the cells have the same genes, but they only use the ones they need. So they get activated differently during development. So a cell that is going to become a vascular cell like xylem in the plant, it activates different genes in that cell than what a cell that's going to become phloem would. So even though the genes are there, that doesn't mean that all of them get used. So that's this differentiation. Then intracellular coordination, this is adjustments of the cell's activity in response to what other cells are doing. So they're not just doing their own thing, they can respond. So for example, when our glucose levels get high in the blood, our cells can respond to that, and therefore they take up the glucose. So they're responding to other cell signals that are in that same multicellular organism. So that's referred to as intracellular coordination. So again, reacting to different hormones. That's a good example. So fungus, of course, are not plants. They have their own kingdom here. There are many distinct kingdoms. There are 74,000 different species. Just a term here, a mycologist. This is a scientist who studies fungi. And again, some of the significant differences here between plants and fungi. Fungi are heterotrophs. Again, we talked about some of these terms. Again, this means that they can't make their own food. They need organic material for food. They form filamentous bodies. They have non-motile sperm. The cell wall they have is not made up of cellulose. It's made up of a carbohydrate called chitin. And they have nuclear mitosis. They form these structures unique to fungi called hyphae and mycelia. So the fungi have these slender filaments called hyphae or hypha for singular. And they're essentially basically long strings of cells, and different ones associate with each other to form these large structures. And the large structures or a mass of these hyphae are then called mycelium, or mycelia is plural. So the main body of the fungus is actually not the mushroom, but instead an extensive network of all these fine, small, thin hyphae that are down in the soil or in the wood from which the fungus is growing. So kind of like a tree, the roots of that would be this mycelium made up of all these hyphae. So the mushroom is actually a very small part of the entire organism. And again, they can have meters of these hyphae. They can go on and on and on. This is just showing an example of this. So again, kind of looks like a spider web almost, but just these long, thin strands of hyphae collectively making up this big mycelia. They also have what's referred to as a septa. So these fungal cells, the way they can intercommunicate or talk to each other is that typically the cells are separated incompletely by this septa. So it's not a complete division of the cell wall, but instead where they can kind of mix their information. So the cytoplasm flows between the cells of the hyphae through these little gaps called septa. There can be many nuclei that are connected together, sharing that same cytoplasm. So the protein synthesis throughout that can be carried to the tips, kind of like our neurons, where the neurotransmitter is made up in the cell body and it travels all the way to the terminals. Well, these proteins within the hyphae can be transported to the ends. And all parts of that fungal body are metabolically active. So they're all going through metabolism. And this is just showing, again, the septum. So you can see here where the cell wall starts growing, but it stops and it leaves this opening here called the septa. Now, reproduction and nutrition, they can reproduce both sexually and asexually. So all fungal nuclei except for the zygote are haploid. Often sexual reproduction is different, has different mating types, must participate. So for example, when two hyphae of different mating types come in contact, then the hyphae can fuse. So again, they don't necessarily fuse with each other. They require different hyphae, different mating types in order for that reproduction to occur. A couple of terms here that we'll talk about. So dikaryotic. So we have these fungi where the nuclei of two fused hyphae do not fuse immediately, but they kind of coexist in the cytoplasm. So at this point, again, there are two, so it's called dikaryotic. So these hyphae have two nuclei in them. So if the nuclei are from genetically different individuals, so like two different types of molds or fungi, that is called a heterokaryon, so hetero meaning different. If the nuclei are genetically similar, so from two of the same types of fungi in those hyphae, then they're called homokaryons. So again, dikaryotic just means there are two nuclei. Again, whenever those fuse together, they both had their nuclei. Now they're both in this same fused cell. If they're from similar individuals, homokaryon, different, hetero. Now, some of the terms that go along with the reproductive structures. So when these structures do form in fungi, a complete septa separate the reproductive cells from the rest of the fungal body. So they are separated. They're not fused and connected where their cytoplasm runs together. These reproductive cells are isolated. So there are three kinds of these structures. The gametangia forms haploid gametes. So we think about our gametes, gametangia. So again, these are haploid. Sporangia are haploid spores. We'll talk more about them. And then conididophores are asexual spores or conida. So again, haploid gametes, we have haploid spores, and then we have asexual spores. So we'll be using these terms as we go through the different types of fungi. Well, gamete was a familiar term to us because that's what our cells use, egg and sperm. Spores are kind of unique. So spores are a common mean for reproduction in fungi. So these are different than like the endospores of bacteria, that dormant state. So these spores are reproductive. They're meant to allow for reproduction. They are well suited for the needs of the organism that are anchored in one place. For example, like ferns use spores a lot. So they're small and light, so they can be suspended in the air, so they can be carried great distances by the wind. When a spore lands in a suitable environment, then it starts germinating. It begins to divide, and now we have a new fungus here. So these spores are a way to allow dispersal of the organism. Another unique part to fungi is that they have external digestion. We, of course, have internal. We have a stomach and intestines that break down all of our food. But for fungi, they secrete those same or similar digestive enzymes to break down, lipids or proteins or carbs. They release them outside of their organisms, and they absorb back into their bodies as smaller organic molecules. So for example, many of them can have the enzyme cellulase to break down cellulose, the cell wall of plants, that releases glucose, that then they absorb the glucose into that organism. And some of them are even carnivores, meaning they can eat meat. So for example, they release proteases. The oyster fungus attracts nematode worms, and then they actually break down those worms and feed on them. All right, there are 8 different fungal phyla that are distinguished from each other. So we will be going through each of these, all of them except the Zygomytica are monophyletic. All right, so here's a nice little summary table. I recommend, making note cards or looking at this, or, just kind of taking one side and covering the other, so that you can tell the difference between these. So first of all, starting with the Microsporidia, so spore, thinking about sporidia or spore forming, they are intracellular parasites of animals. They actually used to be considered protists, but then were classified because they are some of the smallest known eukaryotes. So they, again, thought to be a protist, but more information determined that they are actually a fungus due to the presence of some mitochondria. There's also the Blastocladiomycota. Whew, these are fun words to say. All right, so these are flagellated gametes. So they're called zoospores. So those gametes, again, haploid, they are flagellated though. And they alternate between haploid and diploid generations. So a lot of these differ based on their reproductive strategy. Another fun one to say here, so we have Neocalamastomycota. So these also form those zoospores, but they have numerous flagella on them. They do not have mitochondria though, so they are obligate anaerobes. They cannot be in the presence of oxygen or it'll kill them because, again, they don't have mitochondria. And these are typically found in the digestive tract of herbivores, so like our deer and other grazing animals. The Chytridomycota, these also produce flagellated gametes, so zoospores, but they're predominantly aquatic. So most of them live in water, freshwater. Some of them are marine though. Then the Zygomycota, these reproduce sexually and asexually. They have multinucleate hyphae, but they don't have that septa except for the reproductive structures. So it's the fusion of that hyphae that leads to the formation of the zygote, where we get that diploid forming. So those in which the meiosis occurs just before it germinates. Then the Glomeromycota, these have large non-flagellated multinuclear asexual spores. This is from symbiotic mycorrhizal associations. We'll come back and talk about what those are, but they interact with different plants to form these mycorrhiza, and they both benefit each other. So this is involved though and helps with their reproduction. Our typical mushrooms that we think about, those are the Basidiomycota. They reproduce sexually. They're born on this club-shaped structure. Again, that mushroom is called a basidia. They have a terminal hyphal cell that produces spores for them, and that spore-producing cell is called the basidium. So we have the basidia, which is the mushroom type structure, and the basidium, which produces those spores. And then occasionally they can have an asexual reproduction. And then our morels that are typically edible mushrooms, they are the Asamoccata. They reproduce by sexual means. Those ascospores are formed inside a sac called the ascus. And then they do commonly have an asexual reproductive method as well. And not that you'll have to know the numbers, but just to give you an idea, those are the most common. So about 32,000 different species. Again, these are the morels that are typically edible, but definitely not all of them. Now, looking at some of the ecological roles and the importance of these fungi, So fungi and bacteria are the principal decomposers. So these are what we need to break down all the dead stuff, all the dead leaves that fall, the dead animals, the dead organisms. They recycle those so that we can reuse those nutrients. Fungi are virtually the only organisms capable of breaking down lichen that's in wood. So again, those decomposers are vital to helping us rid of dead material. And by breaking down the substances, again, they release those building blocks. They release amino acids, they release carbohydrates, and make them available to other organisms. Unfortunately, there are some, just like our prokaryotes, that cause disease, though many less than our prokaryotic friends we talked about last chapter. So these fungi can have these disease-causing organisms. Again, they can infect both plants and animals. So a lot of crop destruction, a lot of trees and forests have been essentially wiped out by different fungal diseases. The world's actually largest organism is an Armillaria. So commercial uses of the fungi. So they have many important uses. They're used yeast, of course, which is one of the only single cellular organisms. It's used to make bread, to make that bread dough raise. Of course, our alcoholic drinks like beer and wine, they're added to different cheese, soy sauce. All of those depend on fungi to produce them. Lots of antibiotics. So penicillin, the first antibiotic, came accidentally from a fungus. And some of them can actually, just like the bioremediation we talked about with prokaryotes, help clean up toxic compounds in our environment. Of course, some of them are edible, not all of them. So some of these edible mushrooms, like we commonly put in salads, but others are not and can be very poisonous when eaten. Again, back to these symbiotic relationships. So there are two main types that we will talk about, but some of them have different symbiotic relationships, just meaning that both organisms benefit, but they typically have those with algae and plants. So the fungus helps the other organism absorb minerals. So all those hyphae that are extending out, they help absorb different minerals and nutrients that are provided to the algae or to the plant. Again, the photosynthesizers, though the algae or the plants, of course, then provide them with organic molecules. So they can use the sun, they're autotrophs, but they provide those organic materials for the heterotrophic fungi. Again, these mutualisms, which is whenever both of them benefit. So there are two types of these that are especially ecologically important. The mycorrhiza, we mentioned a little bit earlier. This is between the roots of plants and a fungi. And then we also have lichens, which are between the fungi and either the algae or the cyanobacteria. Now, these mycorrhiza actually occur in about 80% of all kinds of plants. So these are very common. Many plants use them. Again, they benefit by increasing the surface area of the roots. They increase that absorption of different nutrients. And of course, again, the plant gives the food that the fungus needs to go through that heterotrophic metabolism. Again, this is just showing these mycorrhiza and how they interact with the plants to create this mutualistic relationship. Then our other symbiotic relationship here, these lichens, again, this is between the fungus and its photosynthetic partner, typically the green algae or cyanobacteria. Typically what you see though is the fungus, but then interwoven, the parts you really can't see between the hyphae are those cyanobacteria and the blue-green algae, or sometimes even both. And they can invade harsh environments, but they are very sensitive to pollutants. And then just a picture here of the rock with a bunch of lichen on it. Again, mainly what you're seeing here is that fungus, but what you can't see are all of those cyanobacteria or the blue-green algae that are within those hyphae.